All right, well, good afternoon, everybody. Thank you so much for joining on a holiday today. So I want to talk to you about a NIFWIF-funded project that I headed during my time at Texas A&M University. And this was a collaborative project between Texas Parks and Wildlife, the Texas A&M Natural Resources Institute, and the Bioinnovation Lab out of Kennesaw State University. So the objective of this project was to look at the feasibility of various treatment options for the deadly bat disease white nose syndrome and the extent to which these treatments and our white nose management actions could result in disturbance to affected bat species. And in this case, we're talking about the tricolored bat or Perimyotis subflavus. So first off, I just want to give you an introduction to white nose syndrome in case it's something that is new to you. So white nose syndrome is a disease caused by the cold-loving fungus Pseudogymnoascus destructans. And I'll generally be referring to this as PD or P destructans throughout the talk. So once the fungus reaches high enough loads on an infected bat, the fungus can be visibly observed on the nose and also the wings and forearms of the bat. And infected bats show abnormal winter behavior, so they awaken from hibernation more regularly. This significantly depletes their energy reserves and results in evaporative water loss. So there's a high mortality rate in these affected bats, and some roosts have actually been entirely wiped out due to white nose syndrome. Now, it's important to note that the presence of the fungus doesn't mean that white nose syndrome has or will develop. So first, white nose syndrome only develops in hibernating bat species that enter a state of torpor. So in torpor, the bat's body temperature decreases to near ambient temperature in order to conserve energy in the winter months. And it makes the torpid bat suitable as a host for the fungus to develop. And if it develops in high enough loads, that's when white nose syndrome can present. However, in some cases, the fungus doesn't get to high enough loads that white nose syndrome occurs. Sometimes, particularly in the first year of infection, you won't see the white nose syndrome occurring from the PD infection, but then the following year, you may see white nose syndrome develop and start seeing that die off. So other species of bat can act as vectors for the fungus, but they don't develop white nose syndrome because of their ecology and behavior. So for instance, Mexican free-tail bats can carry the fungus, but they don't develop white nose syndrome. Throughout the country, the most affected species by white nose syndrome are the northern long-eared bat, the little brown bat, and the tricolored bat. And the fungus can be transmitted from bat to bat, and also from environment to bat as well. So also understanding the fungus reservoirs essentially in the substrate where bats are roosting is also an important factor when it comes to white nose syndrome. So white nose syndrome was first detected in the winter of 2006 in New York State. And since then, the disease has spread across much of the United States and into several provinces in Canada as well. So there are 12 species of bat that have been recorded as developing white nose syndrome. And there's a further 8 species that have tested positive for the fungus, but don't exhibit diagnostic symptoms of white nose syndrome. So for instance, like I was saying before, the Mexican free-tail bat. And this map here is from whitenosesyndrome.org. And it's showing the current state of things across the country. And this is a map that's easily accessible online at whitenosesyndrome.org. So I want to take you back to 2018, 2019, when this project first started. So a little bit about the history of white nose syndrome in Texas. So we first detected PD in the Texas Panhandle in early 2017, and it was detected in cave myotis, tricolor bats, and Townsend's big-eared bat. So we've got Townsend's big-eared bat up here at the top. This is one of those species which seems to act as a vector for the fungus, but doesn't seem to develop white nose syndrome, whereas both cave myotis and tricolor bats have developed white nose syndrome. And in 2018, the fungus continued to spread into central Texas, and it was further detected on Mexican free-tailed bats. So that's where we were at the beginning of this project. And we're going to be talking about the tricolored bat today. So one of the first things to note about tricolored bats is that they are very, very cute, and I call them furry chicken nuggets. They're one of the smallest species of bats that we have here in the US, and they've been documented in all the vegetative regions of Texas except for the far western portions of the Trans-Pecos, the sort of the El Paso area. And it's most common in the eastern half and central part of the state. So they're a hibernating bat species, and they're commonly found in caves in winter, but in East Texas, they have been found overwintering in large numbers in culverts. So there are approximately 17 culverts there within a stretch of highway, where we have counted up to 3,000 bats within these culverts. So these culvert roosting bats were first identified in the literature by Sandel et al. In 1996, and TPWD and Texas A&M University have been monitoring these culverts since the winter of 2016-2017. The culverts themselves are all concrete box culverts. They run along the width of the highway, and they have a lot of substrate over the top of them. They have water in or around them, because culverts are obviously there to act as a way to move water away from roadways. And some of these culverts have several sections. So when I talk about a culvert having like 2 sections, essentially that's like 2 culverts directly next to one another. And this region has no natural karst areas. So there are no local caves in this area. The one that we know that's nearest is actually over 100 miles away. And like I said, these can house really large numbers of tricolor bats. We also find them in clusters like this, which is very rare for the species. They're a species that usually roost singularly and not in clusters like this. And these culverts exclusively house tricolor bats. So in a couple of the culverts, you might find the occasional southeastern myotis or big brown bat. But in terms of actually like hibernating populations, we only find tricolor bats in there. So as of 2018, when we started this, we had 1 positive swab from a bat for the P. destructans fungus. Bear in mind, at these culverts, I take nearly 400 swabs from the substrate and bats. So we just have the one positive one. But the problem is that population models show that even with a treatment of really high efficacy, if it's applied after the initial declines have occurred in that roost, it would require many more decades for populations to recover. So bats, for being as small as they are, they're long-lived. They have a low reproductive rate, usually 1 pup a year. Some species have more, but usually 1 pup a year. And in 2018, experts came together to discuss potential proactive treatment options to protect this super important population of tricolor bats from white nose syndrome in Texas. So the aim of the project was to minimize the impact of white nose syndrome at no tricolor bat culvert roosts in East Texas by applying white nose treatment options before and during the initial invasion of the PD fungus. So Texas Parks and Wildlife, Kennesaw State University, and Texas A&M University came together to trial 2 treatment options of these culverts. The first was polyethylene glycol, or PEG. This is applied to culvert walls and ceilings during the summer. So this is a treatment option that is treating the hibernacula and not the bat. The other are volatile organic compounds, or VOCs. And this is applied to both hibernating bats and culverts. So this is applied in winter, and essentially bats and the roosts themselves are kind of fogged with these VOCs. So my main role in this project was to look at how logistically feasible it was in this environment to be able to conduct these treatments at culvert hibernacula. One of the good things with culverts is that we don't have those really sensitive cave ecosystems that you're going to be finding in a lot of places where bats are hibernating. You know, we don't have any like rare or endemic species that are specific to a culvert under the I-45. So it allows us a greater range of treatment options to trial. So I wanted to see how logistically feasible it was to do this. And we also wanted to know how much disturbance is being caused to the tricolour bats through our management actions. And by that, I mean not just applying the treatments, but you know, our surveying efforts and also our swabbing efforts to keep up with the presence of the fungus in the environment. So I'm going to talk to you a bit about how we applied the treatments and what we learned from this. Okay, so I'm not going to go too deep diving into the treatments and the development of the treatments. So this was sort of Kennesaw State University's main role in the project. And it was decided that PEG 8000 and VOCs were two of the best treatment options that we could trial. So PEG is a polyether compound, and it has an ability to inhibit growth and germination of PD. So the idea is that you apply PEG to the hibernacula substrates before the bats arrive for winter, and it makes the hibernacula a more inhospitable environment for the fungus. So you're likely to reduce viable fungal loads and therefore reduce infection and disease severity. VOCs, volatile organic compounds, they're low molecular weight, low solubility in water, and have a high vapor pressure, so they readily evaporate from liquid to gas. We used a combination of flavazon 185B combined with decanol, and these inhibit fungal growth both on the bats and also within the hibernacula as well. So we split up our culverts into essentially treated and untreated culverts. So because it was an integrative disease management project, the type of treatments applied each year were open to change based on our findings and on the logistical feasibility of the treatments. So we actually did treatments over 4 years. And you can see the red and blue, those are our treatment culverts, so those first 6. And then we have 3 control and 4 management. Now, the only difference with the management from the control is just that they had much larger groups of bats, and essentially we weren't willing to be doing anything with these larger populations until we knew more about the treatment and if it caused any sort of level of disturbance. So generally we can refer to these as treatment, so the VOC and PEG culverts, and untreated, so the control and management culverts. And you can see in the first year of the project, we applied VOC to 3 of them, PEG to 3 of them. The next 2 years, the culverts received both PEG and VOC treatment, and in our final year, just the VOC treatment. And you can see we have the number of sections here. So down here, for instance, TAMU 28, this is our super duper large culvert, which has actually got 6 sections. So it's essentially 6 culverts stacked next to each other. And we have counted up to 1,000 tricolour bats roosting in there. Okay, so let's talk about the PEG treatment. So first things first, this was taking place in summer, and we needed to remove any wildlife that was present in the culverts prior to us actually applying the treatment. Now, for as many bats as we get here during winter, in summer, it's maybe a couple or they're not there at all. So for the very few bats that did need to be removed, we put them in a cloth bag and kept them in an air-conditioned car so that they didn't overheat whilst we applied the treatment, and then they released back afterwards. So the amount of PEG that we needed was calculated based on the dimensions of our culverts, and this was done by our microbiologist at Kennesaw State. Oh, bear with me a second. All right. So what we would do is we would mix up the peg in the field. We would essentially mix this powder with the water to create the amount that had been calculated in the field. We then poured it into a 2-gallon reservoir of a gas-powered backpack sprayer. And then if there was any remaining peg solution that was left that needed to be done, it was poured in, it was distributed between 1-gallon buckets. So we then loaded all these items into a garden cart. You can see me, I'm sitting in it down there. That was on the front slide as well. I'm sitting in it with the backpack sprayer on me. And we attached the garden cart to a winch, and then we pushed the cart with everything in it through the culvert. And what we would do, because that culvert was going to be cart was going to be winched back through, we would drop the buckets with the extra peg where we thought we needed them in the culvert, where we believe we would need to actually be refilling the reservoir. So yeah, the personnel to apply the treatment, we call them the applicator. They pulled the culvert all the way through, dropped those buckets off. And upon starting the sprayer, the winch operator then pulled the cart back through at a steady speed whilst the applicator applied the peg solution. So we used sort of a sweeping back and forth motion from left to right in order to cover the walls and the ceiling. The annoying thing was when the applicator actually had to refill the reservoir, we would have to listen to hear them stop the motor, then we'd have to stop the winch, wait for them to fill back up, and then get the motor started up again to start spraying again. So it required quite a bit of focus and cooperation there to make sure that that was getting done. The success of the peg application varied between the culverts, and it was influenced by several factors. So culvert water levels, mud, and other sort of debris could cause some issues. So several of the culverts showed significant buildup of mud and sediment, which made access through the entirety of the culvert quite difficult. And sometimes it actually prevented the applicator from moving through the culvert because the winch just couldn't pull it through that mud. In one case, there was a culvert that had so much sediment buildup at the entrances that we were actually unable to get the cart in. We actually had to purchase a couple of manual backpack sprayers and have a couple of us go in there and someone else walking along behind, sort of dragging the rest of the peg in the buckets. So that was definitely challenging for sure. In other cases, we had debris like metal posts or concrete posts or branches or something like that. And these would have to be removed before we could try and get the cart through as well. And there was just some difficulties where the cart wouldn't stay sort of on track and they'd get stuck and they'd have to get out and shift it so that we could continue winching it through. Now, the height of the culvert was also really important with this as well, because with the smaller ones, we needed to use the winch, but we did have a couple where they were tall enough that someone could actually hold onto the cart and pull through. And you can see that on this photo on the far right, and that's Jonah actually pulling the cart with our applicator on there through the culvert. And we found that this provided a steadier and more consistent speed and a more consistent application of the peg solution as well. Okay, so now let's have a chat about VOC. So we had two things that needed to be calculated for the application of the VOC. We needed the amount needed and the speed at which the applicator has to pass through the culvert in order to achieve a concentration of 500 parts per million. Once again, our amazing microbiologist calculated all of this, and he developed this somewhat automated aerosolizer. So the cart had the measured chemicals, it had a pump, it had a fan to assist with actually pushing out and spreading the VOCs. And we were able to attach this to a winch, which had a speed control. So we were able to pull this through at a rate where we were able to reach those concentrations. And we also didn't need somebody in this. So essentially what we would do we would have this connected to a winch. Someone would push it all the way through the end of the culvert, and then they would block up the end of that culvert with just some plastic sheeting. And that was to make sure that we could keep the chemicals inside and they weren't drifting away, because we wanted to make sure we were reaching the required concentration. We would then block off the end with the where the cart would be coming back through, so where the winch was. Again, just to kind of keep that in, making sure we're reaching the right concentrations. And we would be able to turn it on with a remote control that Dr. Gabriel had created. So that was great. We could turn it on with the remote control. It would be winched through at a consistent rate that had been calculated. So by the time it reached us at the winch, all the chemicals had been used up. So we'd then keep the sheeting in place just for a little bit of time, and then we would go ahead and remove it. So it was definitely less time consuming and less work than the PEG applications. But the automated cart still had some of the difficulty with the substrate, although not nearly as much, potentially due to being kind of lighter in weight. However, on rare occasions, it did become stuck. Like I said, we would sometimes get like mineral or sediment buildup on the wall. And a couple of times someone had to sort of put on a respirator, run in there, get it unstuck, and then we would have to keep it moving. So how feasible are each of these treatment options? Okay, well, we could do them both, but both came with pros and cons. So definitely PEG took more time and physical exertion than VOC. So partly, of course, I said we had to remove bats, and there weren't very many, but we did have to remove other wildlife prior to the application. And in all instances where this occurred, this was snakes, and then a couple of times it was water moccasins. And it's not fun being in a culvert in a very cramped space with water moccasins. So that was definitely something that we had to consider, although it did actually only happen the one summer. So I'm not sure what was into them that summer. Yeah, and so the individual applying the PEG was required to like push the cart through the entire length of the culvert, move the sprayer, reposition the cart, refill the sprayer, restart the sprayer. And this is all in really cramped conditions. So there was a lot of physical exertion. And bear in mind also this was in summer in Texas. So we're talking pretty difficult conditions to be able to do this. So we needed more personnel in order to be able to do this, just because of the sheer physicality of it. You know, we couldn't have somebody going in and doing 2 of those a day. That being said, it was simple to do. It was very simple to measure out the amount of PEG we needed, mix it up in the field, and do it there and then. It was a relatively easy calculation. The VOC treatment definitely required more specialized components and specialized knowledge in order to have everything ready to go into the field and get it working. So you need to do, you know, more work in the office, more work in the lab before you're able to go out into the field in order for that to work successfully. Certainly the PEG treatment, that was something that we at A&M and TPWD could do by ourselves. But when it came to the VOC treatment, we really needed our microbiologist from Kennesaw State there to oversee things and make sure that they ran properly. I would say one of the more difficult things with both of these was just the application process, and the fact that in some culverts it just wasn't possible to treat them fully or necessarily treat them correctly because of the issues we had with water and mud, etc. So you can actually see in this photo on the bottom right, this was a culvert that we had a lot of issues with, because after the first kind of year or so, it actually flooded quite significantly, and we could only get about half of the way in. You know, we would go in as best we could with the cart, but you know, we couldn't flood it, and it would obviously, the ceiling would get very, very low. So there was only so much we could do there. And I think a future research and development side project would be to look at creating an amphibious application system. So something that is more able to easily move like over water and land. But overall, I think most of us who are out in the field would say that VOC was a easier option than PEG. Okay, so for disturbance to bats, we looked at several different measures. So we wanted to look at short-term disturbance, and through that we looked at acoustic data. So we placed acoustic detectors outside of the culverts a couple of weeks prior to swabbing, and then VOC, and then after VOC they were removed. And we also looked at bat counts before and after VOC. So this was basically within winter counts. So we would count the number of bats about 15 days prior to VOC application, and then between 30 and 50 days after VOC application. We also looked at bat counts across the years to look at long-term disturbance. So we had counts from Melissa Meyerhofer's project. She was a PhD student at Texas A&M, and she did a TPWD funded project where she was monitoring these culverts. So we actually had numbers dating back to 2016. And finally, we wanted to look at potential resilience to disturbance. One of the big concerns that we have with doing, white nose syndrome treatments is if we're causing disturbance to the bats, we could just actually be exacerbating the issue. We're continuing to wake them when they're already being broken out of torpor more regularly due to the disease itself. And the problem, certainly in with the northern counterparts for the species, is that they're hibernating in areas where there is little to no insect activity in winter. They're not able to replenish their energy reserves. But down here in Texas, we have pretty mild winters, and we have a lot of insect activity, even in winter. So we wanted to see if these guys were actually eating. And if they are eating, what are they eating? Because in that case, it could be that they are able to have some level of resilience to disturbance caused by our management projects, and potentially even some resilience to white nose syndrome itself. So I did a meta-DNA barcoding dietary study, and I've only found one other study that has used this method and looked at winter diets in tricolor bats. And this took place in Tennessee. This is by Bernadette al. Took place in Tennessee. And of all the bats they collected, all the tricolor bats they collected, they were only able to get fecal samples from 8% of them. So they only had 18 samples to analyze. And they found that they mostly ate dipterans, so true flies. But it does suggest that potentially they weren't eating a huge amount, seeing as it was actually quite difficult to get hold of fecal samples. Okay. So these are the acoustic results from 3 of our culverts. The light gray line is temperature, and the dark gray line is nightly activity levels. And this is estimated by the number of tricolor bat calls per night. So you can see the square is where we conducted swabbing. The circle is where we had VOC applications, and the dashed gray line is the mean number of bat calls per night across the period of data collection. So you can see in the case of swabbing, there was one instance where we saw activity levels greater than the mean activity rate at the night after we conducted swabbing, but in 2 others, we actually saw activity below the mean. With the VOC applications, activity levels seem to be very slightly above the mean or on the mean, but honestly, the activity we were seeing was much more closely related to ambient temperature, and you can see that with these R values here. And overall, we didn't see any big spikes in activity after we had been at these culverts. I actually also had these recording during the day as well, because I wanted to see if, potentially we were causing them to actually leave the culvert during daylight hours or something like that. And I did pick up a couple of daytime calls, but it was never when any of the researchers were actually at the culverts. So as for our counts, like I said, we had our pre-VOC counts and our post-VOC counts at our treated culverts. And we did a generalized linear mixed model with Poisson distribution, and it suggested that bat numbers were significantly lower after we conducted the VOC treatment. That being said, I'm going to take this with a pinch of salt, and I want to discuss this more when we get to kind of the discussion part of things. So for long-term disturbance, we actually split up our treated culverts and our untreated culverts and looked at them separately. And this is because, if you remember me saying, the culverts that had the really large numbers of bats were in the untreated section. And because of the big discrepancies in numbers, it made a lot more sense to kind of split them apart and look at them. So we wanted to explore the effects of time in each condition independently, as I said, and we used bat number as our dependent variable, and year was either a factorial or a continuous variable, and then culvert ID was our random factor. So we chose to use time as a continuous variable to examine changes in populations over the entire 7 years for which we had population data, and then we used time as a factorial variable to determine significant differences in bat numbers between each of these years. And then we used, where time is factorial, we used pairwise contrasts. So what we found was, when treated as a continuous variable, so that again is looking at over time as opposed to between years, there was a main effect of time in the untreated culverts, such that the number of tricolour bats decreased over time, but this was not seen in the treated culverts. So you can see the red arrow there is when we actually started the treatments in the treated culverts. And while it looks like there is a decline from 2019 to 2022, this was not statistically significant. However, although it looks less so, apparently there is a significant decline in these untreated culverts. And when we treated time as a factorial variable, there was a main effect of time in both treated and untreated culverts. So essentially that shows that bat numbers do differ significantly across the years. And so we have the letters at the top. So if years do not share a letter, then they are considered significantly different. Now onto the diet. I was thrilled that I was able to get 150 samples, and 131 of these were able to be analyzed. So what I would do is, during the winter period, I would collect bats from each culvert, put them in a little paper bag, and I'd hang them up on a little bat washing line. So what we would do is I would check every 10 minutes. If they produced a fecal sample, they were immediately released. We collected the sample. And if they hadn't produced one after an hour of being outside the culvert, they were just released back in. And using this method, over 60% of captured tricolour bats produced fecal samples. And remember, that's compared to the less than 10% that were able to be collected in Tennessee. Also, December was warmer than January, and we were having a cold spell in January. So it provided the opportunity to look at diet in varying winter temperatures as well. So this is the frequency of occurrence of different groups, different invertebrate groups and families. And you can see there's more species diversity in prey in the December samples versus the January samples. But in both cases, the most commonly consumed order were dipterans, particularly chironomids. And these are a large group of non-biting, primarily aquatic midges. So aside from frequency of occurrence of species, I also wanted to look at composition. So essentially how much of the fecal sample on average is made up of different groups of invertebrates. And as you can see, dipterans made up most of their diet, and particularly in January, where only 1% of their diet was something other than a dipteran, essentially. Okay, so let's have a look at our disturbance evidence. So with the acoustic evidence, we didn't really have any real evidence of an increased activity after the VOC application. The activity that we did record seemed to correlate nicely with temperature, so it's suggesting that we're potentially not really causing all that much disturbance to these guys. Now, something that is important is I don't think we can necessarily assume that these actions don't cause disturbance to all hibernating bats or even all hibernating tricolored bats. These culverts are very loud. They are under a major roadway. You know, you can hear, when you're in there and you hear a truck go over, it's very loud. So there is a chance that the bats that are using these culverts are already somewhat habituated to a certain level of disturbance. And potentially us going in there just doesn't cause them additional disturbance really to what they're actually acclimated to. So I think that's definitely something to keep in mind. Another thing is, for any of you who know about acoustic data collection, trying to collect acoustic data at culverts is a monumental task. There is a lot of interference, the reflective surfaces, the running water. This can often like fill up your SD card, drain your batteries, and also just make it really hard to get nice clean backcalls as well. So the data that I did collect, I had actually tried to collect more from additional culverts, and essentially I just wasn't able to do it just because the you know, batteries or SD cards would just absolutely run out, and I just wasn't able to get the data. So it's definitely kind of more on the preliminary side of things, and I'd love to work at trying to find a better way to collect acoustic data at these culverts. Okay, and our before and after counts. So like I said, we did have significantly smaller numbers before and after our VOC treatments. Now, first off, there was a small effect size of this. So although it was significant in terms of its biological relevance, it's quite low. Now, what we really needed to have to be able to see if this decline was caused due to our treatments is for us to have had those counts at the control culverts as well. We didn't do that because we were worried about disturbing them. So really, it's very difficult for us to pinpoint and say that the reduced numbers we were seeing after VOC was due to the VOC application. It could well be just due to autocorrelation. Essentially, we were getting closer to the time of emergence. Some of these counts were taken a couple of months apart, and you know, these guys leave. Last time I was there, it was kind of mid-March, and most of them were gone. So I really take that with kind of a pinch of salt, and I think we could have done that better. But really, we need to know a lot more about kind of within season movement between and within these culverts as well. We do have evidence from other kind of southern latitudes that tricolor bats will actually move from hibernacula structures such as culverts and caves and actually day roost in trees on occasion as well. So it could be that, you know, when I went back there, they're more likely to be roosting in trees because it's getting slightly warmer and spending less time in the culverts. It's kind of an unknown and a really good area for future research. For our long-term counts, so what it suggested was that there is potentially a decline in our largest overwintering tricolor bat roosts. And this would be due to factors outside of management efforts because we weren't going there to treat them. We would go there once a year just to swab. That being said, it does seem like there is a lot of variability between years because when we treated time as a factorial variable, there was an awful lot of variability there. So I don't see this as like a really huge evident decline or like an indication that there is disease wiping them out or anything like that. I actually think it's probably just kind of like a natural, sort of a natural variation. And by all means, maybe when we get more data next year, we may actually see that correlation removed. So I think that's kind of like a just see how it goes situation. As for the diet, amazingly, 60% of our bats produce fecal samples. So they are out there, they are foraging, they are eating food, and they mostly eat dipterans. That constitutes most of their diet, particularly chironomids. And if I'm saying that wrong, please excuse me. And a little bit of a flash forward to the next slide, but PD was not detected during this winter when I collected these fecal samples. So this activity is not the result of accommodating a disease or PD infection. The interesting thing about chironomids is, based on the reading I've done as I've tried to understand these results, they seem to be more associated with poor quality water or polluted water from urbanized landscape. And obviously that makes a lot of sense. At a culvert, you know, you've got water coming in, culverts are made to have this water kind of wash out away from the roads. It's a very polluted environment. And it seems that potentially these culverts not only provide a really great hibernacula, but they're also providing an abundant source of prey for the bats during winter. Okay, so now a quick talk about our PD monitoring. So as you probably noticed, I didn't talk anything about the efficacy of these treatments, and that is because we were not getting hits in terms of PD infection. So 2019, 2020, we had no positive results. In 2021, we did receive positive results, so 8 culverts, and these included both treated and untreated culverts. We had 8 bat swabs and 3 substrate swabs come back as positive. There were low fungal loads, that's important to note, but they were across like 8 of the 13 culverts. Unfortunately, by the time we received the results and the samples, we weren't able to go back and swab the bats because they had already left in the spring. But our plan was in 2022, we were ready to see PD, we were ready to get swabbing done quickly as possible. And we had no positive results again. Essentially, PD had disappeared. And initially it was kind of a, oh, wow, the treatments are working, this is great. But it also disappeared from non-treated culverts. So I don't think it had anything to do whatsoever with our treatments. So let's have a little look at what we usually see when we look at kind of a PD invasion in North America. So this is from a study by Hoyt et al. It's published in 2020. And you can see that what happens, so this is, we have the bat prevalence and the environmental prevalence. And you can see pre-PD invasion, and then you have this first invasion in year 1. So that would have been when we had those first positive results. And you can see that it looks like PD prevalence increases both in the environment and in bats during the first year. And then the following year, it kind of picks up where it left off from. We weren't seeing that at all. It didn't happen with us at all. Ours disappeared. So very interesting. Definitely need to do some further research looking at that. We have been doing some microclimate work, so kind of watch this space. We've had a fantastic student from Austin Peay University who has been doing some microclimate modeling. Really interesting results, and we'll definitely be having a webinar on that in kind of the next year or so. But very, very interesting that something is going on there, and potentially these hibernacula, these culverts are actually a kind of a refugia for this species. So in conclusion, VOC and PEG can both be applied to culvert hibernacula, and I think the next step would be to create some sort of amphibious application device. Our P destructants monitoring and management efforts do not seem to disturb the bats at these culverts. There's no clear evidence that we're causing them issues, and they are up and active and eating during the winter. And P destructions appeared and then disappeared twice. So it looks like we've had potentially 2 failed invasions. Why this is, we need to figure out. And a good next step would be to look at environmental reservoir research at these culverts as well. And finally, you have many, many people to thank who were involved with this project, both from Texas A&M, Texas Parks and Wildlife, Kennesaw State, support from TxDOT. And I had a lot of students and volunteers come out with me and had the opportunity to come and join me. In fact, one of my students even went and got a bat tattoo after joining me on this. So it's been a fantastic experience, and I want to thank everybody for their support and help.
Thank you, Samantha. So everyone, just as a reminder, the Q&A function is in the lower right-hand corner on your screen, and I will be moderating and asking Dr. Levers the questions from there. So the first question that we had that I did not answer in the chat was if you could provide a few words about the disease in South Texas.
South Texas, we know little to nothing in South Texas right now. I think there have been a couple of swabbing efforts down there. I believe that Victoria, there were some positive samples from there. But in terms of like fatalities from the disease, mortalities from the disease, we haven't received any information about a lot of mortalities, and just generally the monitoring efforts down there have not been as rigorous, because it's less likely that you're going to be getting the hibernating species down there, and certainly with South Texas being even like one of the warmer places as well, they're probably less likely to be affected at this stage.
We had a note from someone with TxDOT asking about their role in this, and I thought it might be worth saying a few words about their participation.
Yeah, so TxDOT have been a great support with us working with these culverts. They've been great at allowing us to go out there, continue to monitor this population, share the data with them, and we've actually had a couple of TxDOT individuals come out and see what we're doing as well, and they've been a fantastic support and really care about the welfare and outcome of these populations.
And then we had another question about potential impacts to the water quality from the VOC or the polyethylene glycol treatments.
Yeah, I know that was definitely of concern. Essentially, all of these projects are considered, was it GRAS? Well, I can't remember what that stands for, Jonah, can you? Generally regarded as safe. There you go. They're generally regarded as safe. So these are all components of things that we find in everyday stuff. So for instance, PEG is like one of the main components like of Miralax. So they're all regarded by the FDA as generally regarded as safe and not having any kind of significant impacts on like the environment or on water quality. In terms of like specific kind of references for that, I would have to go in and dig them out.
Yeah, I can share that we, at the early stages of this, we did some really extensive digging on all of these, and I felt like they were all, and in the quantities being distributed, they were all either the types of chemicals that you know basically break down really quickly in aquatic systems or with sunlight, or are just like with polyethylene glycol, you can literally consume very large amounts of it and be totally fine. So there's a question about, can you clarify the recent Texas Parks and Wildlife Department press release regarding white nose syndrome and the new species it was detected on?
Yeah, sure. So this winter we had several developments, and some of you may have seen the press release. One of which is that we now have confirmed white nose syndrome in tricolored bats in Central Texas. So we had 2 tricolored bats we actually had 3 tricolored bats that presented with visible fungus, and we were able to confirm white nose syndrome in 2 of these bats. So this is the first case of white nose syndrome in tricolor bats in Texas, and it's the most southernmost detection of the disease in this species as well. That being said, it didn't look like it had a particularly significant effect on the population at that site. We didn't see a massive die-off. We haven't had any people kind of calling in and saying they found dead bats and they're tricolor bats. I've also been working with the rabies identification laboratory. When we had a mass die-off of cave myotas in 2020, they had a mass influx of cave myotas carcasses for rabies detection, and we're not seeing anything like that. So essentially, yes, our tricolor bats in Texas do now have white nose syndrome, but the extent to which it's going to affect them is something that is kind of, we don't know yet. It's waiting to be seen. In terms of the new detections, our diversity biologist, Krista Demery, out in the Trans-Pecos, she does our PD monitoring out there. And this year we had several new myotis species test positive for the fungus. So we had Myotis thysinodes, Myotis volans, and we also had canyon bats, Pistralis hesperus, test positive for PD. And this is the first time that species has ever tested positive for PD anywhere in the country. So the other 2 Myotis species, it was the first time in Texas, but for the canyon bat, it was the first time it had ever tested positive anywhere. I hope that clears it up a bit.
So next question, we had some significant winter storms during this time frame. Could these have impacted PD spread and or bat counts since they vary annually?
Yeah, so that's a really good question, because obviously, you know, we definitely see in the media about these significant weather events having a big effect on our bats. And often with these significant weather events, these big freezes, we see a lot of mortality in Mexican free-tail bats. And the reason for that is that these guys aren't built for that. They're not built to withstand those super duper low temperatures. I will say that after our 2021 Snowmageddon, when it was safe, I did actually head out to the culverts to check on the bats. And I absolutely did find some dead tricolor bats, but mostly what I found is that they had moved as much as possible into cracks in the culverts. And there was no real evidence of any kind of significant difference like the following year in the numbers. So there were some fatalities due to those kind of big like break freeze, but nothing that seemed to affect them on like a population level at this site.
There's a question that says, you may have mentioned it already, but what is the mortality rate of bats with white nose syndrome? And I might just be able to add that we know that a very large number of tricolored bats, something like 90% of the population has died, right? In the U.S., in the eastern part of the U.S. where they've been impacted.
Yeah, on like a roost by roost basis, a hibernacula by hibernacula basis, it's usually somewhere between 90 and 100%.
But there's a bunch of species that it doesn't impact, so it's kind of hard to answer that as a general question.
Yeah, and even the species that are impacted can be impacted differently. So, you know, big brown bats, for instance, they do develop white nose syndrome, but I don't believe that they don't die in that level of kind of rate that we see with, say, tricolor bats or little brown bats.
Yeah. So Lois Ballin with Parks and Wildlife asked if there's been any testing in West Texas, and the answer is yes.
Yes.
That's ongoing. Clover Clamens with TxDOT asked, is the lack of a true hibernation / torpor season contribute to potential lack of PD detection?
Yeah, absolutely. That could very well be the case. And it seems to be that there's evidence that these bats are not going into like a deep hibernation. They're in a shallow torpor. They're then coming out of it, feeding, replenishing, and this potentially could mean that they are not going to develop white nose syndrome because of this. That doesn't necessarily mean that PD wouldn't be able to thrive there. We do know from the microclimate work that we have done that the environment is conducive to the development of the fungus and the ability for the fungus to grow. And because we have found it on a couple of occasions, the fungus can definitely be there. But whether it's able to stick around based on the general overall climate of our winters there seems like it may be in question. But certainly, I think even if the bats were infected with PD, potentially their specific hibernation behavior of this low level of torpor and eating is potentially preventing them from developing white nose syndrome. That being said, when we did have the cave myotis die off from white nose syndrome in 2020, the bats that were collected were not emaciated. So they had died from white nose syndrome, but they had also been eating. So again, it's one of those situations where PD and white nose syndrome is so different down in the southeast and in the southwest, and we just still have so much to learn about how it works down here.
Okay, so we are at 1 o'clock, but we have a fair number of questions left. I'm wondering if we can sort of attempt a lightning round of questions and just we'll see how many we can get through in the next 3 or 4 minutes, and then we'll just have to call.
I'll try and answer quickly.
Has the fungus been detected in Houston area bats?
I would need to check the map, but I don't think so.
I recall a free-tailed bat north of Houston, one low-level detection once.
Okay, I
But it
guess.
was a county north. I'd have to look at it too. John Young asked if there's a low risk to water quality, etc. Is there potential treatment in caves that are infected with white nose syndrome? And I'm happy to take this one if you would like, Sam.
Yeah, go for it.
Okay, so my thought on this one is that because of the extremely high endemicism in caves, just the unique endemic organisms that live in a cave, that even though these chemicals or substances are not expected to have really detrimental impacts in spring or aquatic systems, they are antifungal in nature, and some of those systems are so devoid of nutrients that there might be very specific cascades, like trophic cascades and food webs that depend on some kind of fungus that's in that cave. And so it would take a lot of research, I think, for us to feel comfortable that we could go spray it in a native cave ecosystem.
Be concerned about messing up the natural biome of the cave, which is obviously going to be including fungal.
Has there been testing done at Bracken Bat Cave since Mexican freetails are known carriers?
No.
There has been testing done.
There hasn't been testing done. As far as I know, there hasn't been any testing done at Bracken.
Oh, no, there has. Sorry. Yeah, BCI has. We haven't done it, though.
We haven't done it. There you
go. Yeah. What are the plans for using this in a larger treatment effort around the state?
That's still kind of under discussion at the minute. Like I said, the kind of next big thing for me would be to look at creating a device that's better at applying, so like a more amphibious application device. But then it is, again, also in terms of the VOC and the PEG, in terms of the amount of effort that's required and specialized knowledge. So I think that would be more of a conversation, and it would be more collaboration and with TechStop, probably that's not something it's probably something that we would have to create with them.
Yeah, and I'll just add too that this was a really unique situation where we had such large roost sites with a bat species that's known to be susceptible and an artificial roost type that it's hard to know if we have that same scenario where, I mean, it would not be worth it if there was 10 bats in a site to go do this much effort. And so that's my thought too. Is there a specific type of license needed for application of PEG? We did have to go through some significant licensing, but Kennesaw University handled that. I can't recall the details of it. So what is the status of cave myotis in hill country caves?
Good question. So after that mass die-off that we saw in 2020, we haven't since seen any kind of mass, like additional mass mortalities. Obviously, we can only look for places in cave myotis where we know there are. And obviously there's going to be roosts that we don't know about, but we have been monitoring both summer and winter roosts that we do know about. So TPWD have been working alongside BCI to do that. And also since we had that die-off as well, TPWD have been conducting emergence counts at several known summer colonies in Central Texas. And I'm actually going out next week to collect some more data for that. So we're keeping an eye on our colonies, the numbers, seeing if we're seeing further declines, if there's maybe some kind of increase. But it seems like we've probably had that like big mass die-off, and maybe these are the survivors who are going to find their way through it. That's our hope.
So so Clover Clamens and John Young with TxDOT both had questions about future of treatments, either at these same sites or elsewhere. And I think maybe my sense is maybe rather than answer that question here, there was a lot of questions from TxDOT in this that really point to we should probably have a get together internally, and let's get together and talk about some of these plans and brainstorm ideas and stuff together at some point. So I don't mean to skip over those questions, but I think that would be the best approach. I will also say that Jennifer Smith Castro with U.S. Fish and Wildlife pointed out that that the PD positive that was near Houston on the free-tailed bat was in Liberty County in the winter of 2018, late 2018, early 2019.
Thank you, Jennifer.
And then I think that's all we have time for today. Thank you, Sam, a lot again for your time. And before people leave, I have one more little detail to share here.